Hi everyone, welcome back to another video. So today's video is kind of like a part one of, a multi-part series about cell biology, in particular the actual cell. So this one's just going to detail some organelles. You'll notice that USABO doesn't have very many questions that are just targeted at organelles, but it is very important to have a good understanding of them for, to be able to do well in the rest of the exam, just because a lot of the questions do hinge on this information. So yeah. Alright, so first we're going to start by looking at eukaryotic cells. So, if you don't know, there are two kind of broad categories when we're looking at organisms and cells, and that's prokaryotic versus eukaryotic. So every type of cell falls into one of these two categories. We as humans and animals, we're eukaryotes, so are plants, while prokaryotes are like your bacteria, your archaea, right? So let's look at some similarities. One similarity is that both of them have plasma membranes. These are made out of phospholipids, and they just create a separate environment for the cell in the outside environment. Next, both of them have cytosol, which is kind of this like jelly like sort of liquidy thing inside of your cell. So it's like kind of this bluish stuff in this picture right here, and this greenish stuff in this picture right here. So that's just kind of everything that's in the interior of your cell, right? Like all the fluid, not counting kind of all the solid parts, right? So we call that cytosol. And then there's also chromosomes in both of them. So now you'll see there's a difference in how those chromosomes are kind of arranged. So you'll see in the prokaryotic cell, the chromosome, or the DNA, it's kind of this one big circle, and then it's kind of just crammed in there. It's like spaghetti almost. And you'll also find some plasmids, which are these tiny loops of DNA. So that's kind of how DNA is organized in a prokaryotic cell. Now, eukaryotes also have chromosomes, but they're inside this area we call the nucleus, which is what we call an organelle. So yeah. And then lastly, both of them have ribosomes, which are not technically organelles because they do not have a membrane around them. As you'll see, organelles, kind of one of the requirements for something to be an organelle is that it does have a membrane around it. So ribosomes don't, but they are really, really important for life in general since they help make proteins, and thus that is why both prokaryotic and eukaryotic cells kind of need to have them. So yeah, we'll continue on with the differences. So the definition of what the cytoplasm is different between prokaryotic and eukaryotic cells. So you can think of the cytoplasm as basically the cytosol and everything within it. So in prokaryotes, cytoplasm is literally just everything inside here. However, for eukaryotes, it's everything excluding the nucleus and what's inside it. Inside the nucleus, it's often called like karyoplasm. So yeah, although it's basically the same type of stuff, they do make a distinction between cytoplasm out here and karyoplasm inside the nucleus. And then also size, prokaryotic cells are usually like 10 times smaller than eukaryotic cells. And then lastly, eukaryotic cells do have membrane-bound organelles, and this is something that we call compartmentalization, where you have a lot of the same reactions that do the same things happen in the same place, aka in one type of organelle. And this just makes things much more efficient. So yeah. Alright, so now let's talk a little bit about the plasma membrane and cell size. So a future lesson will talk more about the plasma membrane, but this is just kind of an introduction to make the rest of the lesson make sense. So the plasma membrane is a selective barrier that, covers the cell and also some of the organelles within it if it does have organelles. It looks kind of like this. So this is a colorized microscope picture. And you'll see you have your phospholipids, but you've also got a bunch of proteins within it. So, also with outside, like these outside membrane pieces where you've got the phospholipid heads, those are going to be hydrophilic. They're going to like water, whereas the hydrophobic region, that's all lipids, so they're not going to like water. So yeah, And then yeah, these proteins, they're like, a lot of them are like channel proteins. They let things go through this selective membrane. And then when you have a protein like embedded in the plasma membrane, you call that an integral protein. You can see that this protein right here is kind of like hanging off to the side. We call that a peripheral protein. So yeah, and then oftentimes these proteins have like little carbohydrates or something attached to them, and this helps a lot with cell signaling. So that's basically kind of how the plasma membrane looks like. And then yeah, in terms of size, surface area grows more slowly than volume when you increase the size of a cell. And this is a really important concept to look at, um, because it kind of determines how big a cell can be, right? So the surface area is determined by, like, if we just model a cell by a cube, the surface area is determined by the side squared, whereas the volume is the side cubed. And then a high surface area to volume ratio is needed to conduct exchange. Hopefully that makes sense, because the more surface area you have, the more contact you have with the outside environment. So cells can't get too big, or else they don't have a high enough surface area to volume ratio to do all that exchange they need to. However, cells kind of get around this sometimes by making themselves different shapes that maximize surface area. For example, microvilli are attachments on intestinal cells that help you absorb things, and they're literally these like little projections that look like little fingers, and it just maximizes the surface area it has in contact with your intestines, or like the inside contents, and thus you're able to absorb a lot more. So now let's do like a walkthrough through some different eukaryotic cells. So a big one you're going to see is the animal cell. And keep in mind these diagrams are all very generalized. Depending on the type of cell, the type of animal, etc., your cell's going to have very different compositions. But this is a very like comprehensive sort of diagram that shows kind of all the common things that show up in an animal cell. So let's just start from here and kind of work clockwise. So first you've got your nucleus, which is where your chromosomes, your DNA are housed. You've got a nuclear envelope surrounding it, and it's got two membranes. You've got a nucleolus, which is where, like rRNA and ribosome synthesis kind of is concentrated, and then chromatin, which is, DNA and proteins, and they're all in this nucleus. And by the way, I'm going to be going into detail in a lot of these things, later in the slideshow. So this is just an overview. Then you've got your plasma membrane on the outside of your cell, ribosomes, the Golgi apparatus, which is responsible for kind of packaging things that are getting sent into the membrane of the cell or like outside the cell, lysosomes, which help with a lot of like digestion and like waste recycling stuff, the mitochondria, which help with ATP synthesis, peroxisomes, which help with like lots of metabolism and whatnot. And then you can see you've got some microvilli right here that help increase the surface area. And then don't worry about the cytoskeleton yet. There's going to be a lesson coming out soon, kind of dealing with this. And then centrosomes, this is more of like a cytoskeleton microtubule thing again. Don't think too much about it. And same with flagellum. But these are all things that you can find in an animal cell. You've also got the endoplasmic reticulum divided into the rough ER and the smooth ER. We'll get into those. But basically, they do a lot of metabolic things, and the rough ER in particular has a very big role in protein synthesis. And then now we can look at unicellular fungi. These are going to be somewhat similar to animal cells. You can see kind of here's a microscope picture of what it looks like on the outside, and then here's like what it looks like on the inside. See a lot of the same things: a nucleus, a mitochondrion, cell wall, and a vacuole, which we'll get into later. So here's a plant cell. In a lot of ways, you'll see that it's pretty similar to an animal cell, but it's got some distinctive things. So, we've got your nucleus, the ERs, ribosomes, and then something special to plant cells, or at least that animals don't have, is a central vacuole. And what this does, it's got a couple of functions. It stores some enzymes, but mostly it's mostly water. What's nice about this is that means the cell doesn't have to expend its energy to make cytoplasm in order for a plant cell to grow bigger. Basically, it needs to just feed water into the central vacuole, and then the plant cell gets much bigger. And this is a really efficient way for a plant to grow itself, so this is a pretty big mechanism. And then, yeah, chloroplasts are also something that are special to plants. They help with photosynthesis. And then you'll see plasmodesmata. They're these like little holes where like a plant cell's cytoplasm is continuous with another plant cell's cytoplasm. And this is how kind of plant cells do communication with each other and whatnot. You've got a cell wall, which animal cells don't have. It's made out of cellulose, and it just basically makes your plant nice and strong. And yeah, you've got a lot of the same things. And yeah, so that's pretty much what a plant cell is. And then going back to plasmodesmata, the animal cell equivalent to that is called a gap junction. It wouldn't need to go through a cell wall, but it does need to go through the plasma membrane. All right, so now let's look at this unicellular algae. It's going to be pretty similar to a plant cell because plants did come from algae. So you see the nucleus and the nucleolus inside. You got a vacuole, got a chloroplast, you got cell wall, and you've also got some flagella, which are kind of special to this type of algae. So yeah. All right, so now we're going to talk about like specific organelles that are really important. So yeah, I'm just gonna give like a lot of specific information about them. So first, we're starting off with the nucleus. This is the big thing in the middle that has all of your DNA in it, in a eukaryotic cell anyways. The nucleus contains most of the genes you find in a eukaryotic cell. Chloroplast and mitochondria have their own genome, so that makes things a little more complicated, but in general, most of the DNA in a cell is found in the nucleus. The nucleus is surrounded by a nuclear envelope, and it does have two membranes in said envelope. And there's this thing called a pore complex that lines these pores that you have in your nucleus, called nuclear pores. So if you look over here, this is like a very detailed picture of a pore complex. You do not need to know this. I just think it's kind of interesting to look at, And this picture shows like, you've got a nuclear pore here, and then you'll have a pore complex like this around it. And then you've got something called the nuclear lamina on the nuclear side of the envelope. So on this side of the envelope, on the side facing the inside, it's made out of intermediate filaments, and its job is to maintain the nucleus's shapes. You don't need to really worry about what intermediate filaments are yet. This will come in the cytoskeleton lesson soon. And you've also got this thing called a nuclear matrix throughout the inside of your nucleus. Again, this is just mostly to support the structure of the nucleus as a semi-round structure. And then in terms of DNA within your nucleus, it's in the form of chromatin. So chromatin is DNA wrapped around these proteins called histones, and you can think of them as kind of like beads on a string where the string is DNA and histones are the beads, right? And then chromosomes, like the terminology is not super well defined, but basically usually when your chromatin is coiled really tightly into like discrete structures, we're gonna call those chromosomes. And when it's all loose and spaghetti-like, we call that chromatin. But yeah. And then you've also got a nucleolus or nucleoli, depending on the cell. So rRNA is made here. rRNA is a very big component of ribosomes. And proteins are imported from the cytoplasm, and they're assembled with rRNA to make ribosomal subunits, which then leave the nucleus through the nuclear pores, and they make it to the cytoplasm where they become ribosomes. So yeah. So now we're going to talk about ribosomes. Ribosomes are made of rRNAs and protein, and they're not actually organelles because they don't have a membrane. So if you have a cell with lots of protein synthesis, you're going to expect lots of ribosomes to make those proteins and lots of nucleoli to make those ribosomes. Hopefully that makes sense. And then the location of a ribosome kind of determines what type of proteins they're going to be making. So free ribosomes are what we call ribosomes that are just floating in the cytoplasm, and most of their products do function in the cytosol. And bound ribosomes are on the outside of the rough endoplasmic reticulum or the nuclear envelope. You'll learn very soon that the nuclear envelope is actually it flows right into the rough endoplasmic reticulum. So yeah. And then most of the products formed by those bound ribosomes are going to function in membranes or outside of the cell, because the RER or the nuclear envelope, they're really good at inserting things in membranes or kind of letting them go outside of the cell. So yeah, if you look at here, this is the rough ER right here. You see kind of these folds, right? And then there's ribosomes bound to them. So those are your bound ribosomes. And you've also got some floating around. Those are your free ribosomes. So yeah, here's just kind of what ribosomes look like. You've got a large subunit, a small subunit, and here's like kind of the complicated model of what it looks like with all the proteins. So yeah. Now we're going to talk about the endomembrane system and then the organelles within that. So, so here's just some information on what the endomembrane system is. The components are the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, vesicles, and vacuoles, and the plasma membrane. And its purpose is to make proteins, and also to transport said proteins into membranes or out of the cell. There's also some of these organelles that are involved in metabolism and the movement of lipids, as well as detoxifying poisons. So transport throughout the endomembrane system is done either through membranes connecting with each other or through vesicles. So yeah, you'll find out a little bit more about this in the following slides. So now let's talk about the endoplasmic reticulum. It's pretty massive, just in terms of how much membrane there is. It accounts for more than 50% of a cell's membranes, and it's a network of membranous tubules and cisternae, which are sac-like. And you'll notice that the smooth ER tend to be kind of like those tubules you see right here, and the rough ER tends to be more of those sacs, those cisternae. So, the ER lumen, which is kind of the inside of the ER, also called the cisternal space, it's separated from the cytosol, and it's continuous, and the endoplasmic reticulum is continuous with the nuclear membrane. So I don't know if you can see here, but on this diagram, you see how like the double membrane of the nuclear envelope just kind of curves into being the endoplasmic reticulum. So yeah, they're consistent with each other, which is pretty cool. So your two types of endoplasmic reticulum are smooth and rough endoplasmic reticulum. Smooth ER functions in lipid synthesis, such as hormones from steroids. And then it also does some carbohydrate metabolism, detoxification of drugs and poison, specifically through this process called hydroxylation, which is when you add a hydroxyl or an OH to a poison or a toxin, and essentially that just makes it more water soluble, and the more water soluble it is, the easier it is to get flushed out of your system, which is what you want. And sometimes the smooth ER also stores calcium ions, and this helps with muscle contractions. In this case, it's usually called the sarcoplasmic reticulum. All right, so rough ER, it mostly makes secretory proteins, so proteins that are going to be secreted outside of the cell, and this includes glycoproteins, and these proteins, they butt off in transport vesicles. So you can see kind of like here, these vesicles are just butting off from the ER, and it's going to make it to the outside of the cell eventually. So yeah. And it also does some membrane making in the same way, it makes some lipids just like the smooth ER. And then by these vesicles, that's like a really good way for lipids to make it to the membrane. So yeah. Now here's the Golgi apparatus / body / complex. There's different ways you can say it, and they're all correct. It's like the warehouse of the cell. It's like UPS. So in here, like the products from the ER, they get delivered to the Golgi apparatus. And here they're modified, stored, and sent elsewhere. So it's kind of just like, you do some packaging and then you send it somewhere else, right? So it's made of flattened membranous sacs called cisternae, if you remember those. And then there is a very specific directionality that goes with the Golgi apparatus. So there's the cis face, which receives vesicles from the ER. and the transphase, which is on the other side and it sends vesicles elsewhere. So between it coming in and leaving, modifications happen between um, that time. So yeah, and then in addition to that, the Golgi apparatus can also manufacture macromolecules, including pectins for plant cell walls. And in fact, when plants divide, that cell plate that forms in the middle of a plant cell, that comes from the Golgi apparatus. So all of those materials do bud off from the Golgi apparatus. So yeah, it's got a lot of functions in this type of thing. And then there is a very interesting model that's been proposed called the cisternal maturation model, in which the cisternae themselves actually move forwards, as part of the process. And the best estimation for what actually happens is a combination of them staying still and moving forwards. So yeah, here are just some pictures. This is like a microscope picture. This is like a drawn one. All right, so now we'll move on to lysosomes. And essentially what they are is they're a sac of hydrolytic enzymes in a, plasma membrane sac. And the enzymes within a lysosome, they work best in acidic conditions. And lysosomes provide these, acidic conditions. It's very acidic within a lysosome. This is really nice in case a lysosome leaks, because these enzymes, they're only really good at ripping things apart when they're in that acidic environment. But when a lysosome leaks into a cell, which is pretty neutral, generally, then those enzymes aren't going to be able to do anything and they won't rip up the cell. Still, in large numbers, they could potentially, but this is really good, in case just, a few lysosomes leak. So there's this thing called phagocytosis that lysosomes can do. Lysosomes can participate in, and this is really good for some cells when they want food, and it's also good for macrophages, which are a type of immune cell. So yeah, essentially what phagocytosis is you just your cell envelops a particle. It could be a particle of food. It could be your immune cell engulfing some pathogen, some foreign invader to your body. And yeah, so when lysosomes, what they do is they your cell phagocytoses the food, and then the lysosome comes and combines with what we call the food vacuole, which is the vacuole that has the food, and then you make something called a phagolysosome. And then within it, your lysosome enzymes are going to digest the food that you just phagocytosed. So yeah, this is very good for, the cell getting some nourishment. Some animals, like amoebas and stuff, this is kind of the only way they can eat. But for us, as humans, this is a pretty good way to absorb some stuff. So yeah. And then lysosomes also do this thing called autophagy, which is when you've got some organelles, some cell parts that are kind of damaged, and you need to get rid of them, but you also want to recycle their materials. You don't want to be wasteful, So if you see here, you've just got a vesicle surrounding some damaged organelles that was just here before. There's a damaged peroxisome, damaged mitochondria, and the lysosome comes, fuses with this vesicle, and then the lysosome's enzymes digest the contents of the vesicle, and then you can reuse those to build new organelles and whatnot. So yeah, this is very good for, recycling things that are old in your body. And then there's this disease, this genetic disease called Tay-Sachs disease, and the symptoms of it come from missing an enzyme that digests lipids. And what happens is that when your lysosomes don't have that enzyme, those lipids, they don't get digested, they end up building up in the brain, and then that's what causes the symptoms of Tay-Sachs disease. All right, so now we'll move on to vacuoles, and vacuoles are essentially large vesicles, and they come from the ER and the Golgi apparatus. And then, you already heard of the food vacuole. There's also this type of vacuole called a contractile vacuole in unicellular protists that live in fresh water. So maybe you know a little bit about osmosis already. Basically, when you've got a cell, in our case, it's our unicellular protist, and it has a higher salt or something else concentration than the environment around it. The water in the environment around it is going to want to go into the cell, and that's exactly what happens to these protists. So they start swelling with water, and these contractile vacuoles, they just pump that extra water out. So that's their function. And then vacuoles also have some function in hydrolysis in plants and fungi. And then small vacuoles in plants can hold organic compounds like food. They may hold poisons that are meant to, deter herbivores. Like, if an herbivore eats a plant that has these poisons, it's not going to eat it again because that's going to be a very bad experience. And yeah, you've also got your central vacuole in plants, which we briefly mentioned. It's what happens when a bunch of smaller vacuoles coalesce. It's filled with cell sap, which is just lots of inorganic ions like potassium and chloride, along with some other things, some proteins, a lot of water. So yeah, this is the stuff that's really inexpensive to make and makes plants growing fast, kind of possible. Alright, so now let's talk about our mitochondria and chloroplasts. So they're special because they have their own DNA, and in a lot of ways, they seem pretty much like independent from the cell. So here's something called endosymbiont theory, which is the theory of how these incredibly independent organelles came to be. So the theory is that a host cell, which we call a proto-eukaryotic cell, because it's not eukaryotic yet, but it is like the ancestor, engulfed an aerobic prokaryotic cell, and that becomes what we call an endosymbiont. And those cells eventually become, like one type of those cells becomes a mitochondrion, and then like another engulfed, cell becomes the chloroplast, and they have like a mutually beneficial kind of relationship. So there's three big like reasons / like proof for endosymbiont theory. One is that they have like mitochondria and chloroplasts have like a double membrane. They have two membranes. For chloroplasts, they actually have a third membrane inside, which we call the thylakoid membrane. And both organelles have their own ribosomes and circular DNA. So circular DNA is kind of like, that kind of tips us off that they were prokaryotic at one point, and then their own ribosomes just indicates that, again, that it's very much like a prokaryotic cell, and it does a lot of its own things, which is the last reason, that it's autonomous within the cell. Like, they divide on their own, they control how many there are in the cell. So yeah, this is just proof for endosymbiont theory. Alright, so now let's look at the mitochondria. So this is what it looks like. Here's a drawing. Here's like an electron microscope picture. And you'll see that it's got the two membranes, and then you got your inner membrane, which is like very convoluted. That's what we call it when it's all foldy like that. And all these folds are called cristae. You've got your little loops of DNA. You've got some ribosomes within what we call the mitochondrial matrix. And then the space between the two membranes is called the intermembrane space. So yeah. And then what's interesting is they can actually form like a power grid in skeletal muscles, which is what's happening like right here. They can like fuse and then separate. And then like together they can just coordinate like energy making. So yeah, here is that in a euglena. Alright, so chloroplasts are a type of plastid. And then plastids, there's also other types of plastids. Plastids are kind of like loosely defined. They're just kind of like a sac of something. There's a more specific definition of that, but that's basically what they are. Amyloplasts are a type of leucoplasts, which are basically just plastids that are like white. And then amyloplasts store starch. Chromoplasts, they store like a lot of like pigments and stuff. So that's why they're they're colored. So yeah, you see you've got your these little stacks. These stacks are called grana, and each of the little discs within the stacks are called thylakoids, and then they're surrounded by their own membrane. You got some ribosomes floating around in what we call the stroma, which is kind of like its own, like cytosol. Then you've got an inner membrane space as well. We've got some DNA. So yeah, that's basically what a chloroplast is. And then here's some chloroplasts in an algae cell. Alright, so here are peroxisomes, which are not mitochondria or chloroplasts, but they don't really belong in any other category because we don't really know where they came from. So peroxisomes have a single membrane, and they have enzymes which remove hydrogens from stuff and add it to oxygen to make hydrogen peroxide, or H2O2. And peroxisomes also have enzymes like catalase, which neutralize the hydrogen peroxide, because hydrogen peroxide is very bad for you. It literally rips your cells apart. So yeah, it's really good. Like there's experiments people do. If you drip like hydrogen peroxide into blood, like the blood like bubbles and then like just basically instantly neutralizes the hydrogen peroxide, because you have like these enzymes in you that are meant to neutralize hydrogen peroxide. So yeah, it's pretty cool stuff. Looks kind of like this. You see that it's got a very distinct like crystal within it. It's usually urease, which is a type of enzyme. And yeah, so it's got a urease crystal. That's kind of its like most distinctive, physical feature. And then a special type of peroxisome is found in like plant seeds called glyoxysomes, and their purpose is to like cut into the supply of nutrients, the supply of food in the seed, to make the seedling grow. So yeah. All right, thank you so much for listening to this lesson. That's it for today. I really hope this was helpful. Again, you'll see that like this doesn't, like, just pure organelle stuff doesn't really show up on USABO, but it is something that's really nice to know for everything else, because it is definitely a basic skill you have to have. It's helpful for like other stuff like AP Bio and whatnot. It's just something you really need to know for everything bio related. So yeah, please leave a comment or a like if you think this video helped you, and please subscribe for more content. Thank you.